If you sequence the DNA of people living today, you can see that all people outside of Africa carry between 2 and 5% Neanderthal DNA fragments.
Neanderthals' ancient DNA reveals an astonishing connection to modern humanity that scientists have only recently begun to understand.
They basically are the closest species or subspecies of us. How much they were actually different, how much we want to see them as different from us, how we can define being human.
Despite diverging from modern humans around 500,000 years ago, Neanderthals left a genetic legacy that persists in people today. Interestingly, present-day people of non-African ancestry carry approximately 2% of Neanderthal DNA in their genomes. Imagine carrying a secret inheritance from a species that split from us nearly 500,000 years ago, and it's still inside us today. The twist? It's not just a trace. It shaped survival itself. Stick around. This story only gets crazier from here. Understanding Neanderthal DNA. The genetic code extracted from ancient Neanderthal bones provides a remarkable window into the past, offering scientists unprecedented insights into human evolution. Examining this prehistoric DNA requires specialized techniques that have evolved dramatically since the first successful extraction in the 1980s. Neanderthal genetic material shares striking similarities with modern human DNA. When comparing the two genomes, scientists discovered that Neanderthal DNA is 99.7% identical to present-day human DNA. This close genetic relationship helps explain why interbreeding was possible between these two hominin groups. For perspective, both Neanderthal and human genomes are approximately 98.8% identical to chimpanzee DNA, confirming our shared evolutionary history. Despite the overwhelming similarity, the subtle but important differences between Neanderthal and human genomes reveal crucial evolutionary adaptations that shaped our survival and development. Scientists have uncovered several genetic variations unique to modern humans that influence essential biological functions, ranging from cognitive development and skull structure to energy metabolism, skin morphology, and wound healing. These distinctions provide fascinating insight into how Homo sapiens adapted differently from their ancient cousins. For example, specific protein coding genes show remarkable differences. SPAG17, which plays a vital role in sperm flagellum function and reproductive success. PCD16, thought to be involved in wound healing and tissue repair. TTF1, a key regulator of ribosomal gene transcription and cellular activity. And RPTN, which is expressed in the epidermis and contributes to skin physiology. Together, these variations highlight how even minor genetic shifts helped modern humans carve out unique advantages that supported survival, resilience, and eventual dominance. Notably, Neanderthals possessed the FOXP2 gene, often called the language gene, including its promoter regions. This discovery challenges earlier assumptions about Neanderthal cognitive abilities, suggesting they had similar genetic predispositions for language as modern humans. Another fascinating discovery relates to brain development. Neanderthal DNA appears to influence neural connectivity patterns in modern humans who carry it. Individuals with higher percentages of Neanderthal genetic material tend to have increased connectivity in visual processing brain regions, but reduced connectivity in areas involved in social cognition. Additionally, research suggests that Neanderthal genetic influence extends to the cerebellum, which plays roles in attention, emotional regulation, and social cognition. Why was Neanderthal DNA is hard to preserve? The extraction and analysis of Neanderthal genetic material presents extraordinary challenges. First among these is the natural degradation of DNA over time. Ancient Neanderthal specimens typically yield DNA in extremely short fragments, usually only 50 to 100 base pairs in length. Consequently, scientists must piece together countless tiny fragments to reconstruct meaningful genetic sequences. Chemical modifications represent another significant obstacle. Deamination of cytosine to uracil occurs frequently in ancient DNA, creating potentially misleading genetic information. This chemical degradation leads to errors that must be carefully identified and corrected during analysis. Environmental factors also significantly impact DNA preservation. The climate where remains are discovered dramatically affects how much genetic material survives. As Jean-Louis Mergny explains, temperate or cold areas of Europe are more favorable than tropical and humid zones, creating a geographical bias in our genetic samples. This limitation means our understanding of human evolution is potentially skewed toward populations that lived in cooler regions. Contamination presents perhaps the most frustrating challenge. When extracting DNA from Neanderthal remains, scientists typically find that over 90% of recovered genetic material comes from microbial contaminants rather than Neanderthal cells. Moreover, ancient specimens frequently become contaminated with modern human DNA during handling and analysis. Given that the frequency of single-base mismatches between Neanderthal and human genomes is estimated at less than 0.5%, distinguishing contamination from authentic ancient DNA requires extraordinary precision. Long before Svante Pääbo became a household name in paleogenetics, he faced one of the most frustrating hurdles a scientist could imagine. Back in the 1980s, while carefully unwrapping the secrets of Egyptian mummies, he made a startling discovery. The DNA, he thought, belonged to these ancient individuals was actually a tangled mix, contaminated by the very people who had handled the mummies for decades. As Mergny recalls, he realized afterwards that what he had sequenced contained parts of genomes that undoubtedly belonged to people who had manipulated the mummies. Imagine the disappointment of meticulously analyzing 23 different mummies, only to find that the breakthrough lay hidden in just one. It's a story of patience, perseverance, and the uncanny luck that science sometimes demands, and it sets the stage for a revelation that would forever change how we understand human evolution. Stick around, because what comes next will uncover how ancient DNA, piece by painstaking piece, reveals the deepest evolutionary secrets hidden in our very genes. How ancient DNA reveals evolutionary secrets. The development of next-generation sequencing technologies ultimately made Neanderthal genome sequencing possible. In 2010, scientists published the first draft of the Neanderthal genome, revolutionizing our understanding of human evolution. This achievement required analyzing DNA from 21 ancient bones found in Croatia's Vindija cave. Through comparative analysis of Neanderthal and modern human genomes, researchers have uncovered fascinating details about our evolutionary timeline. Current evidence suggests that Neanderthals diverged from the primate line, leading to modern humans approximately 400,000 years ago in Africa. The average divergence of anatomically modern humans and Neanderthal DNA sequences is estimated to have occurred around 825,000 years ago, while the populations separated from each other between 270,000 and 440,000 years ago. Ancient DNA also reveals intriguing details about Neanderthal population dynamics. Analysis of their genetic material indicates they experienced a significant population bottleneck, with their numbers dwindling to perhaps as few as 5,000 individuals between 50,000 and 40,000 years ago. This bottleneck created an overabundance of potentially harmful mutations, likely resulting in reduced reproductive fitness and high rates of recessive diseases in their population. The sequencing of Neanderthal mitochondrial DNA, mtDNA, has provided additional insights. Scientists discovered that Neanderthal mitochondrial DNA separated from human mitochondrial DNA approximately 660,000 years ago. Interestingly, Neanderthals had a significantly greater proportion of non-synonymous to synonymous mutations in their protein-coding mitochondrial genes, possibly indicating reduced purifying selection pressure resulting from their smaller population size. The mitochondrial genome analysis also revealed unique structural features. Jean-Louis Mergny found that Neanderthal mitochondrial DNA formed more complex G-quadruplex structures, 4-stranded secondary structures forming nodes in the genome, than those in modern humans. It turns out these twisted little structures in Neanderthal DNA weren't just fascinating curiosities, they may have been roadblocks, slowing down how efficiently their mitochondria could copy themselves. That might not sound like a big deal, but in evolution, even tiny disadvantages can decide who thrives and who fades into history. Unless, of course, the Neanderthals had some secret advantage, specialized enzymes, hidden efficiencies, something we're only just beginning to imagine. And that's exactly why scientists keep peeling back the layers of this ancient genetic puzzle. Every strand they untangle doesn't just tell us about Neanderthals, it tells us about ourselves. So stay with me, because as we step into what researchers now call the mitochondrial mystery, you'll see how these tiny powerhouses of the cell may hold answers that rewrite the story of human evolution, and you won't want to miss where this trail of evidence leads. The mitochondrial mystery, what mtDNA tells us. Mitochondrial DNA, mtDNA, has unlocked crucial mysteries about Neanderthal origins that nuclear DNA alone couldn't reveal. Unlike nuclear DNA, mitochondrial genetic material passes solely through maternal lineage, providing a unique window into evolutionary history. Now let's delve into Neanderthal mtDNA sequencing breakthroughs. The first successful extraction of Neanderthal mtDNA marked a watershed moment in paleogenetics. In 1997, pioneering scientist Svante Pääbo and his team extracted DNA from a Neanderthal-type specimen discovered in Germany's Feldhofer Cave in 1856. This groundbreaking achievement represented the first glimpse into the genetic makeup of our ancient relatives, revealing a previously unknown mitochondrial DNA sequence from a hypervariable region of the mitochondrial control region. The researchers wisely chose to focus on mitochondrial DNA instead of nuclear DNA for several practical reasons. Primarily, mtDNA exists in far greater quantities, with 500 to 1,000 copies in each cell compared to just 2 copies of nuclear DNA. This abundance significantly increased the likelihood of finding preserved genetic material in ancient specimens. Furthermore, mitochondrial DNA accumulates mutations approximately 10 times faster than nuclear DNA, making it a more precise tool for measuring evolutionary timescales. A decade later, in 2008, Pääbo's team achieved another milestone by publishing the complete Neanderthal mitochondrial genome. This feat utilized high-throughput 454 sequencing technology to analyze DNA extracted from a bone fragment weighing less than 0.3 grams, belonging to a 38,000-year-old Neanderthal from Vindija Cave in Croatia. The project generated 8,341 mtDNA sequences with an average length of 69.4 base pairs, creating a 35-fold coverage of the complete mitochondrial genome. Ultimately, these technological advancements laid the groundwork for sequencing the entire Neanderthal nuclear genome, which was first published in 2010. This progression from mitochondrial DNA to nuclear DNA sequencing fundamentally transformed our understanding of human evolution. Comparative analysis reveals substantial differences between Neanderthal and modern human mitochondrial DNA. The assembled Neanderthal mitochondrial genome contained 206 differences from the human reference sequence, including 195 transitions and 11 transversions. This pattern places Neanderthals at a fascinating intermediate position, 3 times more different from us than we are from each other, yet only half as different as chimpanzees are from us. Based on these differences and using the human-chimpanzee divergence date of 6 to 8 million years ago as calibration, scientists calculated that Neanderthal mitochondrial DNA diverged from modern human mitochondrial DNA between 550,000 and 690,000 years ago. Specifically, a 2008 study estimated this divergence at approximately 660,000 years ago, with a 95% credibility interval of 520,000 to 800,000 years. This time frame is roughly 4 times older than the age of the most recent common ancestor of all modern human mitochondrial DNA. Interestingly, when scientists examined specific proteins encoded by mitochondrial DNA, they found that subunit 2 of cytochrome C oxidase in the mitochondrial electron transport chain had experienced the largest number of amino acid substitutions in human ancestors since separating from Neanderthals. This suggests specific metabolic adaptations occurred in the human lineage after diverging from Neanderthals. Perhaps most significantly, researchers found no evidence of Neanderthal mtDNA in modern human populations. As Paebo's team concluded, the Neanderthals did not contribute to the mitochondrial genome of today's humans. This finding contrasts with nuclear DNA evidence of interbreeding, hinting at complex population dynamics. What did mitochondrial DNA reveal about Neanderthal population size? Well, mitochondrial DNA provides compelling evidence that Neanderthals maintained relatively small population sizes throughout their existence. One clear indicator comes from the ratio of non-synonymous to synonymous mutations in their mitochondrial DNA protein coding genes, which was significantly higher than in other primate lineages. This pattern typically signals reduced purifying selection, a mechanism that normally removes harmful mutations due to smaller population sizes. In 2017, scientists made an extraordinary discovery when they sequenced mtDNA from a Neanderthal femur found in Hohenstein-Stadel Cave near Ulm, Germany. This specimen, dated to approximately 124,000 years ago, contained a previously unknown mitochondrial lineage. The HST mtDNA represents the deepest diverging lineage among Neanderthal mtDNAs discovered to date, separating from other known Neanderthal lineages at least 220,000 years ago. Prior to this discovery, scientists had estimated Neanderthal mtDNA diversity through Watterson's estimator θW, yielding the lowest diversity among compared groups, Neanderthals, Denisovans, and modern humans. However, including the HST specimen almost doubled this estimate to 2.50 times 10 to the power of 3, approaching the value observed in Denisovans, 3.46 times 10 to the power of 3. The HST-mtDNA exhibits an average pairwise nucleotide distance to other Neanderthal mtDNAs of 104 positions, greater than any Denisovan pair, and in the upper range of modern human worldwide pairwise distances. These findings indicate much greater Neanderthal genetic diversity than previously assumed. Nevertheless, Bayesian analysis of mitochondrial DNA sequences reveals a consistent pattern of population decline throughout the Middle and Late Pleistocene, with Neanderthal effective population size reaching its lowest point around 42,000 years ago, shortly before their extinction. Multiple studies suggest specific factors contributing to this population decline. One theory proposes that recurrent glaciations in Eurasia during Neanderthals' approximately 400,000 years of existence led to population reductions. Another study examining mtDNA diversity estimated that the Neanderthal population in Europe may have included fewer than 3,400 females. Remarkably, mitochondrial evidence also hints at an unexpected migration event. Analysis of the HST specimen suggests that between 470,000 and 220,000 years ago, a hominin lineage more closely related to modern humans migrated out of Africa and interbred with Neanderthals already present in Europe. This migration appears to have been small enough not to significantly impact Neanderthals' nuclear DNA, but substantial enough to completely replace their original mitochondrial DNA lineage with one more similar to modern humans. It's at this point that the story takes an unexpected turn. For decades, scientists wrestled with a mystery buried in our own genetic code, a contradiction so strange it almost rewrote everything we thought we knew about human ancestry. On one side, nuclear DNA confidently told us that Neanderthals and Denisovans were sister groups, splitting away from us hundreds of thousands of years ago. But on the other side, mitochondrial DNA painted a completely different picture, one where Neanderthals stood closer to us than to their Denisovan cousins. Two pieces of evidence, both backed by science, yet seemingly at odds with one another. What could possibly explain this? Well, this is where things get fascinating, because the answer doesn't just lie in cold data, but in the hidden story of interbreeding, survival, and ancient lineages colliding. And if you stick with me till the very end, you'll see how this single revelation changed the way we view Neanderthal origins forever. Nuclear DNA and the hidden story of Neanderthal origins. The 2010 sequencing of the first Neanderthal nuclear genome marked a pivotal moment in understanding human evolution, finally giving scientists access to the complete genetic blueprint of our closest extinct relatives. This breakthrough, achieved after years of methodological challenges, opened entirely new avenues for exploring Neanderthal origins and their relationship to modern humans. After successfully sequencing mitochondrial DNA, Svante Pääbo and his team took on the enormous challenge of sequencing the Neanderthal nuclear genome, a task once considered impossible. At his newly established Max Planck Institute in Leipzig, Germany, Pääbo's team steadily improved methods to isolate and analyze DNA from ancient remains. The researchers exploited new technical developments that made DNA sequencing highly efficient and engaged collaborators with expertise in population genetics and advanced sequence analysis. These efforts culminated in 2010 with the publication of the first draft of the Neanderthal nuclear genome. The achievement involved samples from Vindija Cave in Croatia, analyzing a total of 21 ancient bones. To overcome the common challenge of contamination with exogenous DNA, Pääbo's team developed innovative approaches, including using restriction enzymes specific to bacterial DNA, which increased the proportion of Neanderthal DNA in libraries 4- to 6-fold. Accordingly, the analysis revealed relatively few amino acid changes had become fixed in the human genome over hundreds of thousands of years. The most affected genes included SPAG17, crucial for sperm flagellum, PCD16, potentially involved in wound healing, TTF1 regulates ribosomal gene transcription, and RPTN expressed in the epidermis. In essence, this genomic data allowed scientists to estimate that the most recent common ancestor of Neanderthals and Homo sapiens lived approximately 800,000 years ago. The average divergence of modern humans and Neanderthal DNA sequences was estimated to have occurred around 825,000 years ago, while the populations separated from each other 270,000 to 440,000 years ago. Perhaps one of the most surprising discoveries came in 2014 when scientists published the complete genome of a female Neanderthal from the Altai Mountains in Siberia. This individual's genome revealed that her parents were likely half-siblings, indicating high rates of incestuous pairings in her genetic lineage. Scientists remain uncertain whether this pattern resulted from her living in a small, isolated population or if other factors influenced the lineage's inbreeding. In a 2024 study, researchers discovered a Neanderthal individual nicknamed Thorin from Grotte Mandrin in Mediterranean France, whose genome showed a relatively early divergence of approximately 105,000 years with other late Neanderthals. Thorin belonged to a population with a small group size that showed no genetic introgression with other known late European Neanderthals, revealing some 50,000 years of genetic isolation despite living in neighboring regions. For this reason, researchers concluded that Neanderthals generally lived in small, highly inbred, and geographically structured populations. They had a substantially smaller effective population size than any modern human population. As a consequence, weakly deleterious, nearly neutral mutations occurring in Neanderthals would have a decreased probability of loss compared to modern humans, as this evolutionary dynamic would be governed by genetic drift rather than negative selection. Recent research suggests the Neanderthal population size was about 20% lower than previously estimated. This revised smaller estimate suggests that Neanderthals may have disappeared because they were simply absorbed into the modern human population. As one researcher noted, recurrent waves of modern human migrations out of Africa eventually overwhelmed the ability of Neanderthals to remain a distinct population, and they were ultimately just assimilated into the modern human gene pool. But what does that really mean for the story of our closest ancient cousins? How did their lives, struggles, and very essence get woven into ours in ways we're only now beginning to uncover? This is where nuclear DNA comes into play, and it's about to completely reshape how we understand Neanderthals, revealing secrets their bones alone could never tell. Stay with me till the very end, because the answers we uncover don't just rewrite history, they might even change how we see ourselves. How nuclear DNA reshapes our understanding of Neanderthals. Comparative analyses of nuclear DNA have fundamentally changed our understanding of the relationship between Neanderthals and modern humans. One striking discovery showed that DNA sequences from Neanderthals were more similar to sequences from contemporary humans originating from Europe or Asia than to contemporary humans originating from Africa. This pattern indicates that Neanderthals and Homo sapiens interbred during their millennia of coexistence. In the light of this evidence, scientists now know that in modern-day humans with European or Asian descent, approximately 1 to 4% of the genome originates from the Neanderthals. For East Asians specifically, their genomes contain about 20% more Neanderthal genes than Europeans and West Asians. Further analysis has revealed that interbreeding between Neanderthals and modern humans started approximately 50,500 years ago and lasted about 7,000 years, until Neanderthals began to disappear. This genome-based estimate aligns with archaeological evidence that modern humans and Neanderthals lived side by side in Eurasia for between 6,000 and 7,000 years. Additionally, nuclear DNA evidence shows that early modern human samples older than 40,000 years already contained regions completely devoid of Neanderthal genes, called Neanderthal deserts, suggesting these areas formed rapidly after gene flow occurred. Most high-frequency Neanderthal genes found in modern humans relate to immune function, skin pigmentation, and metabolism. Some Neanderthal genes involved in immune system function and skin pigmentation increased in frequency over time, suggesting they provided advantages to human survival. The discovery of nuclear DNA has also revealed unexpected bidirectional genes. Recent research identified regions of human ancestry in Neanderthals, estimating that the Vindija and Altai Neanderthal genomes contain 53.9 megabases, 2.5%, and 80.0 megabytes, 3.7%, of human introgressed sequences, respectively. Scientists found evidence for two distinct epochs of human gene flow into Neanderthals. Soon after this bidirectional gene flow was identified, researchers developed a framework showing that sequences identified as Neanderthal in African individuals are significantly enriched in regions of high heterozygosity in the Neanderthal genome. This observation reveals patterns caused by gene flow from modern humans to Neanderthals and suggests human introgressed sequences caused Neanderthal population size to be overestimated. Essentially, nuclear DNA analysis continues to reshape our understanding of human evolution, showing that at the time when Homo sapiens migrated out of Africa, at least two extinct hominin populations inhabited Eurasia, with Neanderthals in western Eurasia and Denisovans in eastern parts of the continent. As modern humans expanded beyond Africa and migrated eastward, they encountered and interbred with both groups, creating the complex genetic tapestry that defines our species today. Interbreeding with Homo sapiens. Genetic evidence uncovered from ancient remains has resolved a long-standing question about human-Neanderthal relationships, revealing an intricate story of interbreeding that left permanent marks on our genome. First and foremost, this genetic legacy varies considerably across modern human populations. The percentage of Neanderthal DNA in present-day humans follows distinct geographical patterns. People with African ancestry south of the Sahara carry little to none, typically ranging from 0 to 0.3%. In contrast, those with ancestry outside Africa possess approximately 1 to 2% Neanderthal DNA. This percentage rises to 14% in some non-African populations. East Asians particularly stand out, carrying about 20% more Neanderthal genetic material than Europeans and West Asians. Indeed, the distribution pattern extends beyond these broad categories. Cushitic and Semitic populations from the Horn of Africa have approximately 1% Neanderthal DNA. Native Americans and Melanesians show higher Neanderthal admixture than Europeans, and remarkably, up to 50% of the entire Neanderthal genome remains present among people in India. Genomic analysis pinpoints when these ancient encounters occurred. The primary interbreeding between Neanderthals and Homo sapiens happened approximately 47,000 years ago, though estimates range from 45,000 to 49,000 years. This genetic exchange wasn't a single event, but an extended period lasting 6,000 to 7,000 years, aligning with archaeological evidence of cohabitation in Eurasia. Throughout these millennia, small pioneering groups of humans migrating from Africa encountered established Neanderthal populations. Analysis of ancient genomes confirms that this interbreeding benefited early humans adapting to environments outside Africa. Scientists have identified 347 Neanderthal genes at high frequencies in living and early human genomes, particularly those affecting immune function, skin pigmentation, and metabolism. Neanderthal mitochondrial DNA seems to have vanished entirely from modern humans. Stick with me, because uncovering why Neanderthal mtDNA is missing in modern humans will reveal one of the most surprising twists in our evolutionary tale. And you won't want to miss the answer that scientists are still hunting for. Why Neanderthal mitochondrial DNA is missing in modern humans. Despite clear evidence of interbreeding in nuclear DNA, scientists have found no traces of Neanderthal mitochondrial DNA in modern humans. This curious absence suggests successful interbreeding happened primarily between Neanderthal males and modern human females. Several hypotheses explain this pattern. Possibly, Neanderthal mitochondrial DNA carried detrimental mutations leading to extinction of carriers. Alternatively, hybrid offspring born to Neanderthal mothers might have remained in Neanderthal groups, subsequently becoming extinct with them. A third possibility suggests female Neanderthal and male Homo sapiens pairings produced infertile offspring. Nonetheless, recent findings challenge the hypothesized incompatibility, revealing that Neanderthal's Y chromosome was replaced by an extinct lineage of modern human Y chromosome that introgressed into Neanderthals between 100,000 and 370,000 years ago. The story grows more complex with evidence of gene flow among three hominin groups: Neanderthals, Denisovans, and modern humans. While Denisovan-derived ancestry is largely absent from modern populations in Africa, Western Asia, and Europe, it reaches 46% in Oceanian populations. Particularly, Negrito populations in the Philippines show the highest detected Denisovan admixture. Meanwhile, bidirectional gene flow has been confirmed between modern humans and Neanderthals. Scientists estimate Neanderthals carried 2.5 to 3.7% human ancestry in their genomes. This finding requires researchers to revise previous Neanderthal population size estimates downward by approximately 20%. Hence, the interbreeding story reveals multiple chapters spanning hundreds of thousands of years. Recent research identifies two distinct epochs of modern human gene flow into Neanderthals. As a result, all people with genetic roots outside sub-Saharan Africa carry Neanderthal variants, while ancestors of people in Asia also mixed with Denisovans. What Neanderthal genes do in us today. Modern humans carry ancient Neanderthal DNA fragments that actively influence numerous biological functions, revealing how these prehistoric genes continue shaping health and traits thousands of years after Neanderthals disappeared. Throughout human history, Neanderthal immune-related genes provided significant adaptive advantages. Between 2 to 4% of non-African genomes contain Neanderthal DNA, with these genes particularly enriched in innate immunity functions. Remarkably, some Neanderthal variants in the toll, like receptor, TLR, family increase immune reactivity to pathogens. The antiviral OAS gene cluster inherited from Neanderthals reduces the odds of developing severe COVID-19 by 22%, illustrating how ancient DNA continues providing protection against modern viral challenges. Neanderthal ancestry appears commonly in regions affecting skin pigmentation, suggesting these variants helped early humans adapt to cooler Eurasian environments. Genes for keratin filaments, which provide toughness to skin, hair, and nails, show significant Neanderthal enrichment. Moreover, around 40% of the Neanderthal genome can be reconstructed from ancestry present in people today. Interestingly, analysis of Neanderthal DNA revealed connections to modern sleep patterns, mood disorders, and addiction. One surprising discovery showed that approximately 1 in 3 European women carry a Neanderthal variant associated with increased fertility and reduced miscarriage risk. Neanderthal DNA explains approximately 2.03% of depression risk, with some variants increasing susceptibility while others are protective. A Neanderthal variant in P-selectin is strongly associated with hypercoagulable states, potentially helpful against ancient pathogens but problematic in modern environments. Additionally, Neanderthal TLR genes may predispose carriers to allergic reactions. Both Neanderthals and modern humans share identical FOXP2 variants, suggesting this language gene predates our common ancestor from 300,000 to 400,000 years ago. Although FOXP2 influences language development, scientists remain uncertain about Neanderthal verbal communication sophistication. Studies of Neanderthal MC1R genes indicate at least 1% of European Neanderthals likely had red hair. This trait evolved independently in Neanderthals through a mutation not found in approximately 3,700 modern humans analyzed. With this, we have come to the end of this video. Thanks for watching. If you enjoyed this video, do well to like, comment, and subscribe for more content. To enjoy more thrilling stories, click now on the following video that pops up on your screen.